Hello, Dr. Glenka. This is Donovan Lucibello,
And Alec Basterchia.
and we're here to talk to you about the results from our lab experiment exploring the GAPDH genes across different plant species. So here you see a beautiful tertiary structure of a protein that we'd hope to be able to further describe throughout our presentation. So by way of introduction, we were looking at the glyceraldehyde 3-phosphate dehydrogenase, or GAPDH enzyme, and it also is related to the isozymes for GAPC, and there are versions 1 through 7. And these are both represented in the Calvin cycle and the glycolysis cycle for plants. So the wide availability in different samples that we could take was part of our hypothesis. Specifically, we're trying to see if both plant samples would yield GAPDH genes with proper lab technique. So the two plant species that we used was one, Coriandrum sativum, otherwise known as cilantro, which is the proud sponsor of Taco Tuesday, and then the oregano species, Oreganum vulgare. Only one of our samples was able to come all the way through the testing.
Now we'll move on to our methods, starting with DNA extraction. In this, we will prepare the DNA template using spin columns and centrifugation. By doing this, we ground up the plant material, which we chose among the leaves of our plant material. Then we purified the DNA using a silica spin column. We finished it off by eluting the DNA from the spin column. Following our DNA extraction, we move on to our 2 PCR steps and then our electrophoresis gel. For our objectives in this PCR experiment, we would first perform our agarose gel electrophoresis to determine if our first round PCR was successful. Then second, we would clean our initial round PCR samples with exonuclease to remove our first round primers. Then third and finally, we would perform second round PCR using first round PCR products, which would amplify our GAPC genes. To create these PCRs, we first made our master mix. Seen below is the ingredients to our master mix. These would be mixed with the primers and the DNA template. Then we moved on to our thermocycler. Seen below is our agarose gel electrophoresis results. From left to right is our first ladder, then our PGAP, then our Arabidopsis thaliana, oregano, cilantro, and water. Our first two, PGAP and Arabidopsis thaliana, are our positive controls. Our negative control is our water. We use these to compare to see what we did want and what we didn't want. Our lower left hand is our initial product, which our initial product showed less results because in this case it amplified everything. Then in our lower right hand corner we found results with our nested PCR. In our nested PCR, this was more specific to our GAPC, so we see results in both our positive controls and in our oregano. So in this case, we would use our oregano throughout the experiment. Once we have finished our 2 PCR steps, we move on to our ligation and transformation lab. Before we would start our ligation process, we must first blunt the ends of our DNA. People may ask, what is this blunting? It first snips off the ends of the DNA to make it so-called even, so that it can fuse or ligate to the Taq polymerase enzyme. Typically, it is only a single adenosine left hanging over, but in either case, the blunting process breaks the bond to make the ends even. This blunting is done using a proofreading polymerase. As seen below, you can see our master mix that we used for our blunting reaction. Moving on from our blunting reaction, we move on to our ligation. The question is, what happens during this ligation? During our ligation, we created a joint or bond. In the case of DNA, it is the splicing of our product and the plasmid, which is our cloning vector. The plasmid we chose, or also what we know as our cloning vector, is PJET 1.2. PJET 1.2 is a circular plasmid with 2 particular genes of our interest, which we will call our reporter genes. Our first reporter gene is the ampicillin resistant gene. Then we also have our ECO471R gene. This ECO471R gene has a natural break in the middle that allows for it to open up to a linear molecule. Normally, this gene is lethal, so we are going to turn it off. Once our ligation begins, we must understand that it is not very accurate. So we have found what our possible products are. There's 4 possible products. First, our plasmid may ligate to itself with the ECO471R gene. Second, there's a GAPC gene which could ligate to itself without the ampicillin resistance gene. Third is the GAPC gene with the PJET 1.2, which ligated should grow as well. And fourth is our 2 GAPC genes plus PJET 1.2 should grow as well, but this will be twice as long. As we finish with our ligation, we look forward to transformation. In this transformation process, we introduce our ligated product into our E. coli. We have to transform them in such a way to make their cell walls permeable to take up this DNA. To do that, they have to be in a rapid state of division, which is in 24 hours old of the culture. This is the only way to get a plasmid into the cell is when the cell is undeveloped. We enhance this with a heat shock. We call this the acceptance stage. Following this stage, we plate our E. coli on the ampicillin agar. In this case, we should only have the GAPC plus the PJET 1.2. The reason for this is the ampicillin resistant gene. From this ampicillin agar, we pulled 4 colonies and grew them in a culture. Then extracted the plasmid DNA from the colonies. We ran restriction enzymes to cut out the GAPC genes from the restriction site. Following this, we then ran our electrophoresis gel with this DNA, and we will be looking for the 1,000 base pair concentration.
So I'd like to talk some more about the restriction enzyme analysis. Let's get right into it. What's the purpose of a restriction digest? Well, it's to find out if GAPC was actually present in the plasmid. As my colleague said, we're looking for a 1,000 base pair sequence. So what we would expect to see is actually 2 bands separated out, one at 3,000 base pairs and one at 1,000 base pairs, with the latter 1,000 base pairs being our payload, the GAPC gene. What else might we see? Well, as we've heard, there are some mistakes possible where we can have 2 genes liganded together, or perhaps not at all. So we're basically going to run out on gel to find out if the ideal state was reached with the amplification of the 1,000 base pair GAPC. In the sequencing reaction, we only have 1 primer, so they can work in two different directions, forward and in reverse, with the overlap in the middle. So in each of those only have about an 800 base pair reach. So now we need to actually mail off our samples to be sequenced. But when we get them back, what we are looking for is to be able is to put the sequence into a software engine to do the analysis piece. When we shipped them off, they were subject to the Sanger dideoxy sequencing reaction mechanism. To the reactions, and 2, it can be an automated process. So with the Sanger dideoxy sequencing reaction, there are nucleotides added in, just like we would for a PCR, but then there are also the dead end nucleotides. These are the DDNTPs, where they do not have they do not have the proper hook on the 3' end to actually join to the next nucleotide. So the synthesis will essentially stop there. And then if a fluorescent dye is added, that would correspond with the particular nucleotide, then we can actually run that out, and with an optical laser we can sequence that DNA sample using automated methods. Once we get the sample back, run through the Sanger reaction. Then we're able to actually upload a data file. When we import that in and we access our files, we're able to lay them one on top of each other. Looking here at the screen, we highlighted our lowest quality signal for this particular part of the sequence. The cutoff we were using was a quality factor of 20. The next step was to create a contiguous file, or known in the biz as a contig. And you'll see two green arrows pointing to the overlapping sequences of the blue band on the bottom and the red band on the top. Part of the screen, highlighted also in a green box, is the quality factor for the lowest quality base pair in this particular part of the sequence, which is 55, and it is still above our cutoff for 420. Once we were able to build our data set, we were able to execute right from the Codon Aligner software a blast search, and the blast search accesses the NCBI GenBake database, which is a publicly sourced database for the scientific community. This gives us the ability to essentially scrub our data against other sequence items done in the past, and we were actually able to bring our hypothesis back in at this point to say that our experiment was moving in the right direction. Keep in mind we're looking at the GAPC gene, and the first search that we highlighted there with the green arrow was the one that we used. Our particular query was 943 base pairs long, and so we were able to correlate that very nicely with the first species to pop up, and that is the Cuphea laiva species. There are some metrics there that I would like to point out to you. One is the query coverage, and that's basically going to tell us what portion of our sequence matched the database sequence. The identity value is going to give you how similar are the sequences in the query region. And the e-value, sort of a weird measure, but it is what probability is that search result likely to be returned with that if we searched a randomly sequenced database or for any other species. Would we get this kind of a match? And so a 0.0 is a very good figure, and that rules out on random hits. So once we were inside the results that we found, and these were put out using the automated features on the blast search, we were able to sort what would be the exons and the introns highlighted there. The benefit of doing this is also in the blast search was that it gave us a sequence of the protein for what all of those triple digit codons equaled out to be. Those are all in the single letter amino acid codes, as you see here. And a rather neat function of being able to get this level of data was to actually put that sequence into the Swiss model engine, and here you can see both the alpha helices and the beta sheets.
As we come into the time of wrapping up our presentation that we spent so much time on, we definitely get a look at our tertiary structure and why this is so important for us to see. We get to see our final results as our almost baby of this whole project throughout the semester. And through using all these different methods to find this amino acid sequence, but also this GAPC sequence that we used for the DNA sequence to send in as Sanger sequencing. So we find that every single method used for our oregano that was, again, our GAPC sequencing that we used was very important in finding our final product and proving that the oregano had the GAPC sequence in it. And our acknowledgements.